have to have same ancestor. Okay, similar functions and similar structure, they are called homologs, and the whole phenomena is homology. Now there is a question. Very similar, I said they have to have a similarity, but how much similar two sequences should be so that you can call them homologs? So generally, if you have a sequence which is more than 100 amino acid long or 100 nucleotides long, then proteins are called homologous if at least 25% of the amino acids are identical. However, you have to bear in mind there are certain known exceptions to this particular rule. For example, there are G protein coupled receptors which are homologous to each other. However, the sequence similarity or sequence identity can be as low as 15% or 12%, but still they are homologous. The other thing is nucleotide sequence. So nucleotide sequences are called homologous if at least 70% of the nucleotides are identical. Then we will say they are homologous. So basically, sequence alignment can give you very good idea about sequence similarity and homology. Now coming on to another very important concept, which is called the e-value. Okay, so what is e-value? We must have heard about this particular thing, e-value. So the e-value is one should not rely on sequence identity alone. Why? You just have to look into this. If you have got two sequences, one is a sequence with 100 residues, and there are 60 residues which are identical in two sequences. And then you have another set of two sequence where you have 200 residues and there are 119 matches. Okay, so 60 residues in 100 or 119 matches over 200 residue. So any person with a biological sense will say yes, the longer sequence makes more sense because it is a longer spread, and longer spread means there could be many other kind of matches. You can have similar kind of motifs, domains, folds, and so on. So longer spread means gives you more confidence that these two proteins could be more similar. However, if we just calculate the percent identity, so the percentage identity, if you go, you will say the first one is probably better because this is showing 60%, the other one will be showing 59.5. Okay. So if you just see the percentage identity, probably you might not be able to make a very good decision. Therefore, in order to incorporate the length of the sequence, there is another concept, the length of the whole amino acids. So there came an important concept which is called the e-value. Okay? E-value full name is the expectation value. So this tells you exactly how likely it is that similarity between your sequence and a database sequence is just due to chance. Okay, so if you have a sequence in hand and if you are seeing there are certain sequences which are similar to your sequence in a database of certain size, then using e-value you can say what are the chance that the sequence is really similar or not. So the lower the e-value, so the lower the e-value means if it is coming closer to 0, so then you can say yes, I am 100% sure that my sequence is similar to the other sequence. So the value which is going close to 0 or the lesser value means that there is an exact match for your sequence in a given database. Okay, so e-value will come later on. Plus this, whenever you blast your sequence, so this comes with an e-value. So what you have to look for, Additionally, the length of the segments between the two sequences, the pattern of amino acid conservations, and the number of insertions and deletions that once you become start doing practice of sequence alignment, sequence similarity search, multiple sequence alignment, protein structure modeling, and all these things, automatically these things will come into your mind that okay, along with the percentage identity, e-value, I have to also see many other things, especially the number of indels and other things. So we will come later on all those things. So coming on to the main thing directly, which is the sequence alignment. So I'm talking about sequence alignment for quite some time, but what exactly sequence alignment is? So sequence alignment is a way of arranging the sequence of DNA, RNA, or protein to identify the regions of similarity between two sequences. Okay. Aligned sequences are typically represented as rows within a matrix. Gaps are inserted between the residues so that identical or similar characters are aligned in successive columns. So if you see here, there are two different sequences, right? One is ending with 882. The other one is 881, or we can say this is 1, the other one is 2. Okay, and then if you have to see without sequence alignment, probably it will take some time for an untrained eye to see the region of similarity. However, using computational tools, you can align the sequences and it will immediately give you this kind of figure. So this shows these two sequences are uh, aligned, right? And the different colors show the different types of amino acids. For example, if you see here the green one, so green one generally depict the hydrophobic amino acids. Okay, and then you have acidic amino acid, negatively charged and positively charged amino acid. Then you have a different kind of polar amino acids and so on. We are here. You can say here this KRR. You know, so these are positively charged. Then you have E, which is negatively charged, hydrophobic, polar, and so on, are the amino acid residues. So if you see here, these two sequences are displayed in such a way that they show the degree of similarity, and the amino acids are placed in successive columns. So if you see here, this particular sequence is somehow aligned, and you can immediately see the level of similarity. Below that, you can see various characters, stars and other characters. So this show the uh, whether the residues are identical or not. So if there is a star, it shows that these two residues are identical. You can see here YECNE, YECN, and here also YECN. So these four residues are identical in these two sequences, and therefore you see there are four stars. If the residue is not similar, then you will see a kind of blank, and if they are somewhat similar, you will see uh, you know uh, two dots, so which shows there is some similarity. Anyway, so we will go on to this similarity thing later on, but first of all, we have to see what this kind of sequence alignment tell you. So if two sequences are derived from same ancestor or they are highly similar, you can see this kind of alignment with very less number of gaps. You can see here only one or two gaps in the entire alignment, and you see there is a high degree of conservation of residues as depicted by different stars. So how can you get this kind of sequence alignment? What are these strategies which are there for sequence alignment? So majorly there are two strategies. One is the global alignment and the local alignment. So global alignment is a coarser methodology that optimizes the alignment between two or more sequence over their entire length. So when it is using entire length, we say the global alignment. When it is using a fixed region, then you can say it is a local alignment. So I will come on to that local alignment thing also. But first we are dealing with global alignment. So the global alignment start at the first residue position and continues in a stepwise manner to the last residue. So the whole length of the protein is examined for the sequence alignment. Regions of the sequences lacking appreciable similarity are aligned to continue the search for similar regions in an attempt to align as much as of the sequences possible. Right. So here one thing is important. Uh, we are looking for more identical residues rather than uh, you know bunch of residues together, which are more which could be more which could tell you greater similarity between two proteins. So basically, this is also said as quantity over quality because we are just looking for more identities, and we generally create more gaps in the alignment. In general, global alignment. But when we go for a local alignment, so the local alignment is sometimes considered very important because there could be regions with highest degree of similarity, and if you go for global alignment, these regions might get bifurcated or diluted. So in global alignment you might miss those regions of important similarities or significant similarities. So what you do is in local alignment, the regions with significant similarity are aligned first in local alignment, which is of course considered a final method, and then you create many regions of optimally aligned residues within the sequences. The alignment of sequences is based on short homologous segments that decrease the number of possible alignments by focusing on the regions that are conserved over evolutionary time. So now you have quality over quantity because now you are looking for short regions with greater quality, right? So we are not concerned only about the quantity. We are also looking what is more logical. Uh, you know, if we consider the evolution. So there are certain regions. Obviously, they will be conserved over evolutionary time because there are certain functions which are conserved, and these functions are related with the conserved amino acids. So those conserved amino acids you can easily find from local alignment. However, as I said, both of the methods provide a reasonable alignment, all kind of possible alignments, but they definitely focus on two different aspects. Now, this is further to explain what is global and what is local alignment. So if you see here, I'm showing you the global alignment on the top. So here, this is the example of global alignment. So here we are looking for end-to-end alignment of the sequence. We are trying to maximize the identity of amino acids in these two sequences. This is the global alignment. But if you see here, we are introducing a lot of gaps in the alignment, which might not be possible if you look from evolutionary point of view. So this is what global alignment is. And as I said, here in global alignment, there is more emphasis on quantity over quality. Now, for the same sequence, if you go for a local alignment, you might see an alignment like this. So here you will see there are two blocks of residues which are aligned. So you can say this looks like more evolutionarily reasonable because a lot of gaps may not be possible in a short evolutionary time, but here you will see a lot of gaps are possible, means inserted. So the local alignment gives you more uh, finer alignment. However, if two sequences are quite similar, you might not see a big difference between global and local alignment methods. Now we are coming on to the sequence alignment strategies. So as I said, the global and local alignments are there. Now, if you go again, so as I said, in global alignment, we have introduced a lot of gaps, or basically we have created a lot of uh, blocks in the second aligned sequence, while in the second one we have created only two blocks, right? So these things, this is to reduce the quality of an alignment. Uh, what we do is there are certain penalties which is called the gap penalties. So if gap penalty means there are penalties for creating gaps. So one possible solution to reduce this creation of gaps is to put a gap penalty for the alignment. So that will discourage the overall creation of gaps, and then you will see less number of gaps as you are seeing it here. So that is you can put a gap penalty, okay, and gap penalty will ensure that you introduce less number of gaps in your alignment. However, it may reduce the quality of an alignment when frequent breaks in the sequence are created. Alignment score assesses which of the many possible alignments are optimal. So there could be many possible alignments which are there. So now, if you compare these two sequences, which I am coming in the next slide, so that alignment scores, they assess which of the possible alignments are the best alignments. Okay. Now, when we perform an alignment, so we look into the methods. What are the methods which we have to choose, especially if you are using a protein structure, a protein sequence, or a nucleotide sequence. So a different kind of alignment tool will be required. Then you have to have a different kind of scoring methods depending upon the kind of sequences and whether you want to assign gap penalties, because as I said, the gap penalties may reduce the quality of an alignment. So we'll take two cases. Case one is high similarity and case two with a low similarity. So the alignment is relatively simple. If the target template sequence identity is above 40%. So if two sequences are uh, identical over 2%, over 40%. Right? There are 40%, let's say for example, amino acids which are identical in given to sequence, then you can say these two sequences are having high degree of similarity, and alignment can be calculated very easily using standard alignment algorithms. So this is one case where you have a high similarity or 40% similarity. Then comes the case where you have low similarity. So if the two sequences, the target template sequence, so the target template sequence is the template on which you are going to align the target sequence. Okay, so if the identity is lower than 40%, right, that means there are amino acids, there are 40% or less amino acids which are identical in these two proteins. And so this alignment will have a lot of gaps, and an expert eye will be required to minimize the number of misaligned residues, or probably you might have to have knowledge of that particular biological system or that protein to see what kind of conserved motifs are there, and you have to properly align everything. So if you go low similarity sequences, or in the case of low similarity, you have to have some kind of manual intervention to improve the proper, to get a proper alignment. Now, alignments also can be improved by including structural information. So this is a very important thing. Uh, if you have a, if you are trying to align a protein sequence, and if you are having a 3D structure of a homologous protein or a similar protein, it could be very beneficial, because you can say I am getting a helix in this particular sequence, so there should not be any gaps in the helix in the other sequence. So in that way the gaps can be avoided in some conserved some given secondary structure elements. in buried regions or between two residues which are far in space. Okay. Now it is important to inspect and edit the alignment in view of the template structure. So this is very important. In case when you have a sequence identity in a lower side or less than 40% or 30%, especially let's say for example in the case of G protein coupled receptors or GPCRs, we have typically low similarity cases where the similarity can be 15%, 20%, and so on. So what you can do is you can add more and more sequences to see if there are some conserved blocks of residues which are present in that. If there are certain given motifs are there, that should be aligned. And if you have some kind of biological insight and you know that this particular amino acid block is required for activity, so those things can be introduced in the alignment as constants to get a better alignment. Now coming on to multiple sequence alignment. So far what we were discussing about the sequence of alignment of only two sequence, right? Now we go for multiple sequence alignment. What happens if you have a multiple sequence to align over each other? So multiple sequence alignment, it is defined as a sequence alignment of three or more biological sequences. Now you have input set of query sequence which are assumed to have an evolutionary relationship. So from MSA, you can infer the sequence homology, and phylogenetic analysis can be done to assess sequence shared evolutionary origins. So multiple sequence alignment is very, very important. It can give you a lot of good information. The basic information MSA provides is the identification of conserved residue, conserved sequence region. So this is the first and the most important thing. Identification of conserved sequence regions, and this is sometimes very, very important, especially when you are dealing with low sequence similarity cases. Now most multiple sequence alignment programs use heuristic methods. Heuristic methods are this kind of learning methods rather than global optimization methods, because identification of the optimal alignment between more than a few sequence of moderate length is computationally quite expensive. Okay. So multiple sequence alignment, it has a lot of strengths. However, it also requires a good amount of computational memory. So what kind of programs are available? So the first one is ClustalW. So ClustalW is a fully automatic program for multiple alignment of DNA and protein sequence, the Clustal program basically. The alignment is progressive and considers the sequence redundancy. So if two sequences are redundant, so they will come into one very easily. Entries can also be calculated from multiple. As I said, one important application of this particular sequence alignment is to identification of phylogenetic or creation of phylogenetic trees. So this is a very important aspect of this particular software, and it can calculate these trees. And another important thing is it has a number of adjustable parameters. However, for the beginners, default values can also be a good starting points. Again, I would like to have a caution here. If you are dealing with sequences which are having low degree of similarity, so how will you know whether two sequences are highly similar or low? Similarity is low. When you do a sequence alignment, automatically it gives you the sequence identity. So looking at that number, whether it is some people say 40%, some people say 30%, but more or less if it is above 40%, you will say highly similar. If it is something lower than 25%, 30%, you will say you have sequences with low sequence identity. So if you are dealing with low sequence identity, you have to see the parameters. You have to tweak the parameters in order to get the optimal sequence alignment. So this is an example of a multiple sequence alignment, and why I am showing you this particular example here is, so this is uh, alignment of multiple sequences from different example different organisms. For example, you have these sequences from bovine, human, mouse, rat, chicken, and so on, different organisms, but they are having a same protein, right? And you see here there are some amino acids, only two amino acids here, which are completely conserved. You see here this K. Uh, you can look at the star and you can say, okay, there are two amino acids which are completely conserved. So this K is completely conserved regardless of the organism. Then you have another residue which is R, which is completely conserved. So what does it tell you? It tells you that perhaps these residues are functionally very important because they are conserved in 100% of the sequences. So that gives you a very good idea about the importance of the residues and importance of the regions as well. For example, if you see here in this particular block, you see there is a lot of green, NT, NT, NT. So if you see, you have a lot of NT in the whole alignment. So you can say, okay, probably this particular type of residue, threonine, and is required at this particular position, and that might have some kind of role. So using this particular multiple sequence alignment, you can also cluster the sequence. So the clustering is, you can see which are the two sequences which are highly related, and then so on. You can say the sequences which are less related, and so you can actually create a phylogenetic tree using this particular multiple sequence alignment, which will tell you uh, some very good information about these proteins, like what are the proteins which are similar to each other, a cluster, which clusters are more similar to the other cluster, and what are the clusters which are very far away from each other. So all these information can be deduced from the multiple sequence alignment, and therefore it is one of the most important tools in you know one of the most important tools in bioinformatic analysis of the protein structures. So, as I said, the MSA is a very important technique, and it can be used for detecting faint similarities in sequences that are not detected by pairwise sequence comparison. If you look into these two sequences, probably you might not be able to see a lot of similarities which are there, which you can see through multiple sequence alignment. Then you can also detect structural homologies. You can group a protein into families. As I said, you look into the multiple sequence alignment, and then you can start grouping protein into different families. The proteins which are similar to each other can be grouped into one family. The proteins similar to the other proteins can be grouped in other families, and then you can see, okay, there are multiple families present in your sequence alignment. So the protein families might have similar kind of functions, right? The proteins in one family will have a kind of similar functions. The proteins in the other family will have more similar functions with each other. And then, obviously, as I said, you can, infer the phylogenetic trees. You can identify the phylogeny among those trees, means among those sequences. You can create phylogenetic trees, and then you can get some evolutionary idea about those sequences. Now, as I said, doing alignment is one thing, but which alignment is the best? So you have to have some kind of numerical criteria, which is called scoring of the alignment. So scoring of the alignment is typically involves construction of a matrix where identical amino acids, or if there are not identical amino acids, the amino acids with similar properties are scored higher than others. So if you go back, so here there are two cases. One is you can say there are amino acids which are completely conserved, marked by a star. So this will be scored very high because it is having uh, complete conservation, right? Then you might see a dot here. So that gives you an idea, a double dot, that you know this particular column is all residues are uh, hydrophobic kind of residue. If you see here, so valine, methionine, and leucine, and so on. So that gives you an idea about uh, the kind of amino acids which are, if they are not identical, if they are having similar kind of properties. So if you score them, you get the second highest score for them, and then you have different amino acids for which you do not give any score. So there are multiple strategies. Of course, there are many people and many strategies have been devised to score the alignments. So the first one is called the identity. So identity shows considering only identical residues, two sequences are aligned, and we say, okay, these two sequences are 40% identical, 20% identical, and so on. And that way we score the alignment. The other one is called the genetic code. It considered a number of base changes in DNA or RNA to uh, interconvert the codon for the amino acids. The third one is chemical similarity. It considers the physicochemical properties. For example, as I showed you, there are amino acids which are, some amino acids are large, some are small, some are basic, some are acidic, some are neutral, and so on. So it the third strategy uses chemical similarity between amino acid, and this is quite a good strategy. And number fourth is observed substitution. This is a very important strategy. So this considered substitution frequencies observed in alignment of sequences. I will come on to those things the coming slides. So this shows how amino acids change over a given period of time, because as time passes by, because of the evolutionary things, amino acids or nucleotide sequences may also change. So this consider the effect of that evolutionary time, and therefore these are considered to be the best methods for scoring of the alignments. So these methods, as I said, based on the alignment, based on the analysis of the frequency which given amino acid is observed to be replaced by other amino acids. So there are two major strategies, which is called the PAM and the Blossom matrix. So the first is the PAM matrix, or percentage amino acid mutation matrix. So this was developed by Dehoff and co-workers, and this gives the probability of one amino acid mutating to a second amino acid within a particular evolutionary time. The scoring schemes are denoted PAM followed by a number, right? So the number would be, the matrix would be PAM 40, PAM 240, and so on. So the lower PAM matrix would recognize short alignments of highly similar sequences. It is important. Short alignments of highly similar sequences, and higher PAM matrix would find longer, weaker local alignment. So PAM 250. So here is the bottom line. If you are using a higher PAM matrix, let's say for example, uh, you know, or let's say if you are using two sequences which might be related, but there is only a weak similarity available. For example, if you are using G protein coupled receptor sequences which are having low degree of similarity with each other, so PAM 250 matrix would be more suitable for the use. The other PAM matrix would be used when you are using sequences would be with high similarity. Okay. Now comes the other one, which is one of the most important substitution matrix, which is called the block substitution matrix. And this was again derived by Dehauf and co-workers based on substitution frequencies from global alignments of very similar. This is important. Global alignment from very similar sequences, and this work was extended by developing substitution matrices using local multiple alignment of more distant local multiple alignment, so creation of blocks. So that's why it is called the block substitution matrix. So basically what happened, a database was assembled that contained multiple alignments without gaps of short regions of related sequences, and these sequences were clustered into groups or blocks based on their similarity at a given threshold value of percentage identity. So the blocks substitution matrix or blossom was derived based on the substitution. So multiple matrix were derived based on a substitution frequency for all pairs of amino acids. Now, the different blossom matrices were obtained by varying threshold. For example, a blossom 62 matrix is derived using a threshold of 62% identity. There would be a blossom 45 matrix, a blossom 80 matrix. So that would be derived using that given threshold. Now coming on to the evolution of the alignment. So now we have done so far pairwise alignment. Then we looked into multiple sequence alignment, the advantages of the alignment. Then we looked into the scoring of the alignment also. Now, finally coming on to this evaluation of the accuracy of alignment. So the best way to assess is to compare alignment from sequence comparison from the alignment of 3D structure. So the easiest way, if you have two protein sequences and you have aligned, whether they are properly aligned or not, you can, if you have 3D structures available, you can superimpose these structures and you can see if there are no gaps in the secondary structures, then or very less or minimal gaps in the secondary structure, then you can say, okay, I have got a reasonably good alignment. But this assessment is only possible if you are working a family of proteins for which you have already known 3D structures. If they are not no known 3D structure, then it would be very difficult for you to do this thing. So for the minimum requirement is you have to have at least two members of the family, and then you can say that okay, I have used multiple sequence alignment, I have got this particular alignment, and as I can compare from the 3D structure, I think the alignment looks good to me. Okay, so that can be a very good strategy. Okay, now comes if you do not have that those kind of things, then you can say there could be some kind of folds and motifs that can be used for the evolution alignment to see whether the alignment is okay correct or not. So as I said, there are multiple tools and techniques for the alignment of sequences. So the first one is BLAST, which probably everybody is aware of, the basic local alignment search tool. This particular tool, BLAST, is used for the initial sequencing, initial screening of the sequence data from any laboratory and to get the idea what kind of protein is there. So for example, you come across a protein which is very new to you, you do not know what this protein is. What you will do is you will try to see whether it is similar to a known protein or not. Okay, and to identify whether the given protein sequence is similar to the other protein, you will use a blast thing. So there are you must be so it blast all these it will blast or it will try to identify similar sequences to your given sequence or sequences, and then you can get an idea like what are the sequences which are similar to my sequences, and then you can also get an idea about their functions. So in short, BLAST search identifies homologous sequences by searching one or more databases. As I said, NCBI. Now, BLAST can be used for a variety of purposes. As I said, BLAST can be used for, first of all, you can identify the species. Looking for a species, a new species. If you're sequencing DNA from unknown species, BLAST may help identify the correct species or homologous species. So this is one very important thing. Then you can also look for protein domains. Those who have experience with the NCBI BLAST, you blast your protein sequence and it gives you a graphical interface where you can see multiple protein domains in your query sequence. Of course, you can look at the phylogeny. You can create a phylogenetic tree or evolutionary tree from the BLAST results. So you can use the sequence similarity among the results, and you can create a phylogenetic tree. So that is also very important. Then comes a different kind of use, which is mapping DNA to a known chromosome. So if you are sequencing a gene from a known species, but you do not know about the chromosome location, so BLAST can help you. BLAST will show you immediately, it will align the given gene onto the chromosome, and you will know what is the coordinates of the given gene on a chromosome. Now, BLAST can also be used to map annotation from one organism to another organism based on the sequence similarity. So, if you are having a gene in one organism, which you know the functions which are used in which are there associated with a given gene, you can also say a similar gene in other organisms also have a similar functions. You can map the annotation from one organism to another organism. So now coming on to the algorithm part, I'll quickly show you this particular part. So basically, BLAST starts with the word of length k, and then alignment is initiated between words of alignment score t. Typically, t would be equal to k, and it starts from multiple positions, and it spread in both of the direction. Okay, and it will see if the regions are getting similar. If you are getting identical amino acids or very similar amino acid, the extension will continue until you get a score which is below statistical threshold. So till then, the alignment extension will happen. So what would be the output? All local alignment with score greater than statistical threshold will be reported. So this is a general BLAST, everybody knows about it. So I'm not going into the details of the algorithm of BLAST, how it goes in both of the direction and so on. Okay, so there are multiple flavors. So different kind of BLAST can be used for a different kind of applications. For example, if you have to compare two protein sequences, you can go for a BLAST P, which is for protein BLAST. Then, if you want to compare a protein sequence with a nucleotide database, so you will use T equals to N. And to compare nucleotide sequences, you will use a different kind of BLAST. Now, as I said, I just touched an important concept which is called the scoring sequence alignment. So, what is the scoring of a sequence alignment? Okay, so here you have two sequences. You see here, there are two sequences. Sequence number 1 is here, sequence number 2 is here. We have done a sequence alignment, and based on that particular sequence alignment, you have got this kind of alignment matrices, alignment matrix. And you can see here there are certain nucleotides which are identical. For example, C is identical to C in the sequence. T in the first sequence is identical to T in second sequences. Similarly, G, A, T are similar to these in the first sequence are identical to the second sequence. However, there is a change. A in the first sequence is not exactly identical to T. So there is a difference. Okay. But there is no gap created. We have not created any gap in the sequence. Gap is denoted by a dash or hyphen. Again, if you look into this CGC, so you have got identical nucleotide CGC, and then you have gap of 3 nucleotides. Okay. So you have here CGT, but no equivalent nucleotides are present here. And then again you have these alignments. So basically the whole idea is, if you look the alignment of these two sequences, you can immediately identify the conserved nucleotides or the nucleotides which are identical. So here you can see this particular block is identical, then you have a different nucleotides. So how can we say this alignment is good or bad? So one way is to see what is the score of the total alignment. So this is, we have a device, let's say there is a general scheme, which shows you have a match score of +1, mismatch score of 0, and you have a gap penalty of -1. Okay. So using a simple scoring scheme, you can actually score the alignment between two nucleotide sequences. For example, you have 18 matches here. So 18 nucleotides which are identical in these two sequences. So we will say the match score is 18 into +1, which is the match score. So that will give you 18 match score. Then you have got two mismatches. Here, this is one mismatch, the other mismatch. So for mismatch, we have a score of 0. So you will see 2 into 0, and then you have a gap penalty. So the gap penalty, as I said, will be introduced if you introduce gap in your sequence alignment. So if you compare, there are total 7 number of gaps which are created while you align these two sequences. So for gap, 7 gaps into -1. -1 is the gap penalty. So the sum of these three will give you the alignment score. So it will lead to 18 plus 0 plus -7. So 18 minus 7, the total score would be +11. So you can say, okay, when these two sequences are aligned in this particular way, I have got a score of +11. Now you change one residue from here to there, you might get a different score. So the uh, hello, is there any question or something? Hello. Okay. So this is how very simple scoring scheme is there, which can be used to score a sequence alignment. Okay, we can skip these things because this would go to you know okay, this will go, and then we'll go into the second part, which is the protein structures in the next part of the talk. So let's quickly look into this origination and length penalties, because that would be very important to understand how we score different kind of alignments. So if you see, there are two alignments, alignment 1 and 2. The same sequences are aligned in two different ways. So this is sequence 1, the other one is sequence 2. Okay, and this is alignment number 1. So this is one way of aligning these two sequences. This is alignment number 2. So the sequences are aligned in a completely different way. Now you might have a question, which one is a better alignment? So in this case, you can see there are two blocks. One block is aligned completely here, then you have a gap, and then you will have another block which is completely aligned. In the second alignment, you will see there are there is a block, then there is a gap, here again gap, then you will have gaps. So there are multiple gaps inserted into the second sequence. Now from the definition, if you go, you will see that which alignment is more likely to be plausible evolutionarily. You can say, yes, it is possible that this particular deletion might have happened, because this is just one change that can happen. That is possible. But to say that multiple deletions have happened would looks, less possible. So alignment number one looks more possible. The possibilities of alignment number one are greater than compared to the alignment number two. So how do we do it in algorithmic way? So we have to devise some kind of scoring patterns, as I have showed you earlier. Now you see here scoring of the sequence alignment, right? So we did a sequence alignment here, and we have a match and mismatch score, as we showed. Match score is +1, mismatch score is 0. And if you originate a gap, you have a penalty of -2, and the length, if it is increased, the length penalty is -1. Now you score the alignment. So as you know, there are 18 nucleotides which are identical in these two sequences. So you will say my match score is 18 into +1, which is your match score. So 18. Then there are two mismatches, so you will get 0 for that. Then there are two gaps which are originated. Okay. So two gaps originated. So for each of the gap, you have a score of -2, and then you have length. So basically, there are total seven gaps. So for each, you will have -1. So basically, the overall score will be 18 plus 0 minus 4 minus 7. So if you say 18 minus 11, the overall score will be +7. So this is how you score this particular given alignment. Okay, now if you go and calculate, what would be the score for the alternate alignment? Okay. So I will stop here. I'll just quickly again say, in order to understand more, you can say you can calculate the alignment score between alignment number 1 and alignment number 2, and automatically you will come to know which alignment is better. So probably everybody will get alignment number 1 is probably getting the better score. Okay, and this is how computational algorithm work. Okay, so this is what part 1 of this particular uh, talk. The other part I would like to show here is, so I think if you have any questions, you can just tell me that whatever you would like to ask me. Now, once we have covered this sequence alignment, I would go on to the protein structure modeling. Okay. So now, why we do protein modeling? So as we have seen that there are techniques of sequences comparison. So we frequently get multiple sequences through laboratory experiments and sequencing, other kind of sequencing studies. So there are two things come to our mind. Number one is, what is the function of this particular protein, and what is the 3D structure of this protein. Okay, because if you know the 3D structure of a protein, you can do a lot of things. You can compare the protein structure. You can see if a small molecule can go and bind into that particular protein structure. So it is also having use in drug design and discovery. You can also find if it is having some kind of interaction partners, other proteins which can interact with this particular protein. So that is a very, very important aspect of any given experimental study. Nowadays, the protein interaction studies are very important. So if you know the 3D structure of a given protein, so you can actually identify those areas where these two proteins can interact, and whether two proteins interact or not. Both of the things you can calculate. So what happens? Why do we do a computational protein structure modeling? Because there are majority of sequences do not have a three-dimensional structure. Okay, I'll show you. Uh, this is an old data, I guess. But if you see here, there are millions of sequences in RefSeq database. However, only a small, minuscule percentage of the entries are there in the protein data bank. Less than 1%. So if you have 99% sequences for which there is no known 3D structure, so obviously you can understand the importance of protein structure modeling, computational protein structure modeling. Okay. So protein structure modeling, what is protein structure modeling? So we try to determine 3D structure of a protein from their amino acid sequence. And I said it is very, very important. It is a very, very important aspect of computational bioinformatics study. Okay. So what we are going to do here is, because these predictive methods, because we do not have more protein structures available from the protein data bank and other databases. So what we try to do is we try to predict the 3D structure of a protein. So these are called the predictive methods or theoretical, and the models which are generated are called the predicted models or theoretical protein models. And this can give you a very good idea about the 3D arrangement of the residues, the secondary structures, the arrangement of secondary structures, the tertiary structures, and the quaternary. A lot of information it can give you about the protein, and it can help you in the design of experiments further. Now, why this particular task is quite difficult? Because the first thing is number of possible structures that proteins may possess is extremely large, as highlighted by the Levinthal's paradox. I will not go into this Levinthal's paradox, but I can just give you an example. Because of the inherent flexibility, when two amino acids join with each other, that gives rise to the possibility of enormous number of structures, because one protein might have a number of amino acids. Say for example, 100 amino acid, 200 amino acid, or a mid-sized protein might have 800 amino acid, and each amino acid has a lot of degree of flexibility. So you can calculate what could be the possible number of structures which are possible. Okay, now the primary sequence may not fully specify the tertiary structure, and direct simulation of protein folding is not generally possible for practical and theoretical reasons. So it is a difficult task. It's not an easy thing to do, but a lot of progress has been made in computational protein structure methods. So basically, there are three main methods which we can use for computational structure predictions. So the first one is called the hello comparative modeling, or homology modeling. So in this, target sequence is used to identify a suitable 3D structural template based on their evolutionary relationship. Structural models can be generated by copying the framework. So basically, first step is you have to identify a 3D template. Then what you can do is structural models can be generated by copying the framework of the template, or you can see the skeleton of the template is copied and then side chains are grown. So in that way you can create a 3D structure of another protein. This method generally results in models with good resolution, which can be used for ligand receptor interaction studies or in silico virtual screening. So this gives you a very good model. If you have the only prerequisite for this particular method is you have to have a homologous protein. So this is number one. When you have a homologous protein, the creation of a 3D structure is very easy. Now, it is also possible that protein from different evolutionary origins might have similar structures, but you cannot capture this thing by traditional homology modeling methods. So in that, what we do is we do a technique which is called threading. So threading tries to match target sequence directly onto other sold structures to identify similar protein folds that can be utilized to build a 3D model. So if there are not direct amino acids, identical amino acids, still using threading you can identify, you try to identify similar protein folds that can be utilized to create a whole 3D structure of your target sequence. Now comes the third one. The third case is much more difficult, because in first you have a template, so modeling is easy. In the second, you can actually you can use threading to match your target sequence on other solved structures. The third is the most important and the most difficult, when both of the two conditions are not there. You do not have any template and you cannot match your target sequence directly onto other structures. So then this is called the ab initio modeling or template free modeling. Ab initio because you are starting from the scratch. So ab initio. So it uses computational algorithms to build protein structure models from scratch, just from using the amino acid. It uses the laws of physics and thermodynamics to see if it can create a structure with least thermodynamic energy. In other words, it will try to create a structure which is the most stable structure from a given amino acid sequence, because it is known that these protein structures are at their minimum energy or highly thermodynamically stable state. So the algorithms try to identify those states using different algorithms. However, because of various complexities, this particular method is limited to smaller proteins up to 100 amino acids, and these uh, methods, because they cannot give you an accurate atomic level model, so they can be used to identify what kind of topology a particular protein is having, or a kind of family or superfamily assignment. So if you are looking for a drug design or discovery kind of project, you have to look for comparative modeling or homology modeling, uh, because it can give you that kind of structure. The other methods may not be able to give you the best kind of structure which are required for drug design and discovery kind of studies. So we will go for the homology modeling thing. So the principle behind homology modeling is the assumption that the structure is highly conserved, more highly conserved than the sequence is, and the functional domains of the proteins are actually structurally conserved. So the conserved domains are transferred from one organisms to another organisms with the sequences. Of course, certain insertions and deletions will be there, but the domains which are responsible for certain functions will be conserved, and the whole idea of homology modeling depends on this particular principle. Now, every protein with a known 3D structure, all the proteins which are available in protein data bank, may serve as a template to predict the structure of a homologous protein. This is a very important uh, thing, homologous protein. So every protein with a 3D structure can predict the structure of a homologous protein. Or in other words, if you say, if you have got a sequence, then you should look for a homologous protein which is having a 3D structure, because that particular structure can be used for the structure prediction of a new protein or new sequence. Now, as I said earlier, if you uh, if you get a sequence identity over 40%, which is called the high sequence identity, then the alignment and the modeling is very easy. If it goes below 30%, then use of multiple templates and the specialized techniques may be required, because you go on to the low similarity zone. So this particular graph, if you look into this, uh, if you go for more than 40%, then no, sorry, if you go for more than 40%, so they are highly similar sequences. Modeling is easy. If it is less than 30%, the modeling would be difficult and would require multiple templates and other things. Now, so you create a homology model, but since it is depending upon your template, so it will look like very similarly. It will look very similar to the template one. Okay, so you cannot study conformational changes just using homology modeling. You cannot see new binding site, because the binding sites which are present in the template will be copied into this, so it will be very difficult to find a new binding site. And then obviously, as I said, because it is created through the skeleton of the template, so there would be obviously large bias towards the structure of the template, and models cannot be docked together. So these are the major limitations. And now the general steps in homology modeling. So these are the general steps. The first one is you have to identify the protein sequence um, which you want to model. Once you identify your protein sequence, then you have to compare this sequence with a database of sequences derived from the PDB. Okay, and to do that, you use BLAST. As I said earlier, that BLAST can be used to identify similarities between two proteins. So you blast your sequence against protein data bank, because it is uh, repository of 3D structure. So you need a 3D structure to create your own homology model. Okay, so when you do a blast search, you have to have sequences with a very low e-value. E-value I explained. So the e-value should be very low. So if your e-value is less than 0.001, you may consider that these two sequences are matching with each other. Okay, and a template with a poor e-value should not be chosen, even if it is only one available, because it will not be the correct template. So the e-value should be less. That should be chosen. Now, if you see here, this is this particular figure tells you the comparative efficiency of generation of homology model. If you look from a target template identity standpoint, so if it is more than 60%, you can say immediately you can create a 3D structure of your target protein, and then you can go for the design and discovery kind of projects. The other thing is between 30 to 60%. So you can say, okay, you can use them for crystallographic applications, and also you can see for site-directed mutagenesis, because you will know the presence and absence of secondary structures, helix and loops and other things that can be used for the planning of other experiments. However, if you are in this zone 0 to 30%, it is a difficult zone, low sequence identity zone. So the functions or the applications would be different. Especially you can go for refining NMR structures, you can go for identification of active site and so on. And also by looking at the fold, you can say, okay, what kind of function it might have. So depending upon the target sequence similarity, you can say the kind of the use of the generated homology models. So as I said, sequence identity is very important. However, template quality, because the sequence is going to be modeled on the template. So the quality of template obviously becomes very important. So you have to select the best template, and this is a crucial choice. This can be a very, very important choice. So what would be the best template ID? So you have two templates. One is 93% identity. The other template is having 90% identity. The identity is not very different, 90 and 93%. But template 1 is having 3.5 angstrom resolution. Template 2 is having 1.5 angstrom resolution. So this tells how good the atoms are resolved in a given crystal structure. So the lesser this number is, the better the template is. So if I say 1.5 angstrom, it is certainly better than 3.5 angstrom, or even 1 angstrom resolution would be better than all. So in my case, I will go for template number 2 because it is having much better resolution and almost equivalent 93% versus 90% sequence identity. So there is not much difference. This is again to emphasize the importance of the resolution. If you see here, the difference between a resolution of 1 angstrom and the resolution of 4 angstrom, you can see all the atomic positions are clearly definable within 1 angstrom, very, very clear. But
Hello, sir. Hello. Hello, sir. Am I audible?
Nikita, Nikita का I think he has lost connectivity.
क्योंकि उनका कुछ भी नहीं है ना यहाँ पे.
He has gone out of it.
उनका कनेक्टिविटी गया होगा. चंदे को कॉल करके पूछना.
हाँ बोलो बोलो.
Video ready है? Question लगाए.
You can write the question in the chat box.
नहीं है उसी में ready.
किसी ने पूछा आपको ये दे जाएगा. किसी ने क्वेश्चन पूछा तो लिख के आपको दे देगा.
अच्छा अच्छा.
हेलो.
नवदीप सिंह.
कनेक्टिविटी के लिए
बैटरी सब नहीं है. हाँ, इस जरा बगल फोन करो. Hello, Dr. Dikshit. I think he's lost his connectivity, so we'll just wait for a few minutes and get back to you all.
He's visible.
Huh? He is visible, but we can't see his mic and video on as such. So there is some connectivity issue from his side. We are calling him back.
Oh, sure, sure.
Officer.
हे नाही ना सब्जेक्ट हाँ
Subject.
ऑथरायझेशन काय ऑथराइज ऑफिसर हे DHL लॉजिस्टिक
Nikita, unmute yourself.
Yes, sir. There is one question. Am I audible, sir? Hello, Dr. Dikshit. Can you hear me?
ये किया था मैसेज किया था
अच्छा,
हो सकता है आवाज नहीं आ रही है
तो फिर कैसे करेंगे?
मैडम आप लेकिन उनको तो वो बोल नहीं पाएंगे तो यू कैन राइट इन चैट बॉक्स एटलीस्ट
तो ये जरा लिख के बोलो।
यू कैन आस्क ना
Chat.
आप बोलो नहीं
Catch it.
Yeah, he cannot hear. You have got Dr. Dixit's number? So we can phone him and tell him this is the question. डाला है? गया ना? हाँ बस चलो. Done. Lecture is over.
Hello, Shailesh.
हाँ सर. Hello.
Hello.
हाँ sir.
Yes. Yeah, what's the strategy now?
Strategy sir has put on chat that you can ask the questions on chat box. Currently he is not
Okay,
getting the
okay.
connectivity over. So he has almost finished it. Just 1 or 2 slides were remaining. Okay. So
Okay.
he has asked for if any questions, then they can put it on the chat box.
Right. Okay. Sure. Okay. Otherwise, we can ask the Madam to offer the chairman's remarks.
Yeah. So Nikita is having some questions she has put up.
Okay. Sure. Sure. Okay.
So like, I don't know, I think we can, I think all the participants, I think they can, will provide sir's email address and they can directly get back to them, right?
Sure. Yeah.
Sir, Hmm. नहीं तो अमोल ने सबको कांटेक्ट किया है, तो वो एक ही लैब में बैठे हैं.
So hello everyone. Actually, somebody is asking email ID of the sir. So his email ID is already given on the brochure. Okay, so you can collect from there, and whatever the questions are there, you can ask directly to him. Otherwise, those questions who are which are there in the chat box, so we will send the email to him, and whatever his replies will be there, so it will be posted in the group. So you can see your answers on the group itself. We will post the answers in the group. So if more questions are there, you all of you are requested to send it on the chat box. We will collect those questions and we will send to him. Thank you.
Dr. Vaskar, can you hear me?
Yes, ma'am.
Dr. Vaskar, then what should we do? Should we conclude the session?
Yeah, I think so. You please for your
Okay.
remarks because yes, sure, please.
Okay. So, from the NIAP project, the PI and all the other organizing committee, I thank Dr. Dikshit for his very informative and detailed lecture on protein sequence and structure analysis using bioinformatics. I'm sure that all the staff and participants have been enlightened today on the topics covering sequencing, alignment, phylogenetic analysis, identifying the correct protein sequences, how to perform the modeling, creating 3D protein structures, crystallographic resolutions in several protein functions. So sir has completed all this in great detail, and I thank you very much, sir, for enlightening all of us. From the team of NIAP and my co-chairman, Dr. Vaskar, and rapporteur, Dr. Shukla, I declare that the session is closed for today. I thank you all for your active participation. Thank you very much.
Participants, tomorrow's session 6 baje hai na? Yeah, tomorrow's session will start at 6 o'clock. So just kindly go through the brochure again because the speakers are from Pennsylvania University. So according to that timing, we have adjusted this time. So we will send you again the link. This new link will be sent tomorrow morning, and according to that, we will join by 6 o'clock tomorrow. Okay. Thank you.